Hello and welcome. I'm Parker Ellingson, an application scientist with Axion Biosystems, and today I'm going to discuss a multiplexed neuronal function assay for assessment of neuroactive compounds. Here we use primary rat cortical neurons cultured in a 24-well plate and demonstrate how phenotypic changes can be detected in structure and functional activity. The Maestro family of benchtop systems are designed to accommodate multi-well microelectrode array plates for label-free, non-invasive, continuous recording from electrically active cells. We plate neurons onto the electrode array and record extracellular voltage, which allows for the detection of action potentials, slow local field potentials, and analysis of network-level activity. The Maestro can provide a number of metrics to characterize neuronal activity. And for this assay, we will focus on four major concepts rooted in the physiology of dissociated neural cultures. Activity is a general measure of excitability and represents the ability of neurons in the culture to repetitively generate action potentials. As a descriptor for activity, we use the mean firing rate of the culture. Synchrony is measured using the area under the normalized cross-correlation, and it represents the ability of neurons in the culture to form synapses and fire together. Oscillations are measured using the interspike interval coefficient of variation, and it is a metric representative of the burstiness or the tendency of the network to organize into bursts. Networks with a proper balance of excitation and inhibition alternate between periods of quiescence and organized bursting. When assessing the effects of a compound on neuronal activity, it's also important to consider the general health of the cell. With a viability tool, the Maestro can use impedance-based technology to assess the coverage and integrity of cell membranes over an electrode. Over the course of 3 weeks, rat cortical cultures displayed increases in activity, synchrony, and oscillations, indicating drastic changes in activity phenotype as the culture matures. Viability remained constant, indicating that the culture had physically changed very little, in that cells are all in the same place and membrane integrity is intact. Day in vitro 14 showed strong, consistent activity with lower variability than DIV-21, indicating that dosing from DIV-14 would give a wider range in which these metrics could be shifted, and, due to the lower variability, will be more likely to resolve differences in dosing response. We dosed DIV-14 primary rat cortical neurons with 4 drugs with distinct mechanisms of action and compared the functional activity to no treatment and vehicle controls. While the drugs we chose tend to be those that increase excitability in a network, the way in which that manifests in network activity may be different depending on the mechanism. 4AP, a potassium channel blocker, caused an increase in mean firing rate, an increase in synchrony, and an increase in oscillations. Picrotoxin, a GABA antagonist, also increased activity and synchrony, but did not have much of an impact on oscillations. Carbachol, a cholinergic agonist, increases activity and oscillations, but induces a small decrease in synchrony. And finally, glutamate, a common excitatory neurotransmitter, increases mean firing rate, but has a sharply negative impact on synchrony and oscillation, suggesting more overall individual activity, but less organized. In conclusion, the Maestro multi-well MEA platform enables high-throughput functional characterization of neural activity. Rat cortical neurons cultured on multi-well MEA plates develop more complex patterns of activity over time. Phenotypes of bursting activity brought about by maturation or dosing with neuroactive compounds can be distinguished using a simple subset of neural metrics, which describe a culture's activity, synchrony, and oscillations. In addition, cell viability can be used to monitor culture health or identify cytotoxic effects of compounds. Thank you for your attention, and we'll see you at SFN 2023.